So second factor, we know that short chain and long chain fatty acids, right? So speaking about short chain, I'll just write down the short chain here. Short chain fatty acid. This is Fa, fatty acid, and here I'll just write down long chain fatty acid. Long chain fatty acid. So you need to compare both short chain and long chain fatty acids. You'll understand. So in previous slide, what we have discussed is temperature is directly proportional to fluidity. Now, this also says that if interaction is more, if interaction between molecules are more than fluidity will be less. Keep this in mind. So if interaction is more, fluidity will be less. That means if interaction is less, fluidity will be more. Keep this in mind. If lesser the interaction, greater the fluidity. Greater the interaction, lesser the fluidity. That's what the concept, right? Because we know that interactions between two phospholipids, if it is very tight, it means fluidity will be less, rigidity will be more. If it is loose, then interaction will become very less and fluidity will become more. This is the general point you need to remember in this case. This is the same principle we are going to apply for all the concepts now. Understood now? So try to understand. Interaction, if it is more, lesser the fluidity. If interaction between the phospholipids is very less, greater the fluidity. Keep this in mind, right? Now, moving further, we'll speak about short chain and long chain fatty acids. Now, so I'll just change the color so that it's easy to distinguish, right? Now, I have short chain fatty acids like this. I'll just write it like this. And long chain fatty acids, I'll just take black color. So these are long chain fatty acids. That means he's speaking about the fatty acid hydrocarbon tail length. About tail length he is speaking about. See that red color tails, you have shorter tails, whereas in case of long chain you have longer chains, right? So these long chain and short chain is important factor which is going to determine the fluidity. So how? We'll see it. So I'll just explain this. So between two, I'll write down 1 2 3 4. So these are the four phospholipids here, short chain, and 1 2 3 4 in case of long chain fatty acids. Between 1 and 2 phospholipid there is an interaction, right? So the interaction is like this, right? Now the same interaction between long chain 1 and 2 phospholipids, see here, see, it's more the interaction, right? We know the general concept. More the interaction, yes, fluidity will be less. That means larger or long chain fatty acids will have more surface area to interact with each other. Hence what will happen, there will be greater the interaction. That means interaction, in terms of interactions between two phospholipids, interaction is less in case of short chain. In case of long chain, what happens? Interaction is more. So due to this, interaction is less and more, fluidity is going to vary. So what do you think where fluidity will be more now? So where interaction will be less, fluidity will be more. It's inversely proportional, right? So in previous only, in this slide only we have discussed, right? Now, so the same concept. So short chain fatty acids, if they are more in the bilayer, it is going to cause more fluidity. Hence high fluidity. High fluidity. Now, in case of long chain fatty acids, what happens? In case of long chain fatty acids, if long chain fatty acids are more, interaction between the phospholipid tails is going to become more. So when interaction is more, it is clearly known that when interaction is more, it causes, yes, that's the right answer. It hence less fluidity. Less fluidity. So this is the concept. So that means short chain fatty acids increases the fluidity of the membrane. Short chain fatty acids increases it. Long chain fatty acids are going to decrease the fluidity of the membrane. Keep this in mind. All the interaction funda is there here. You have to think about interactions between the molecules, the major molecules, phospholipids. So if you think basic like this, then only you can answer it very easily, right? Because it's confusing. Next. So third one, very important thing which we discussed was, we understood about temperature, we have understood about short chain and long chain. Now third important factor is saturated and unsaturated fatty acids, right? So saturated and unsaturated fatty acids plays an important role in membrane fluidity. Again, we know that already, right? So saturated, will consider first one as saturated, and not saturated, it's saturated, and second one is unsaturated, right? Okay. Now, so we know that saturated and unsaturated, we have two phospholipids. I will make it four, which are linear structures of phospholipid tails, because it is saturated. Saturated means single bonded. Always remember, saturated meaning single bonded phospholipid. Unsaturated meaning double bonded unsaturated fatty acids. Now, so unsaturated fatty acids which are double bonded in nature, if I write the structure, so it will be like this. So all unsaturated fatty acids will be like this. Now, so we know that interactions plays an important role, right? So check out about interaction. So it will be like this. Here the interactions will be there. Now it is, in case of unsaturated, what happens here, the distance between two phospholipids are more. In this case, here the interaction is more, but here again it is less. Here again it is very less, and here more, and again less. That means when you look into the interactions, saturated fatty acid tails, these are rigid, rigid, non-flexible, non-flexible, a rod-like appearance, a rod-like appearance, right? Now, because of this rigidity, a rod-like appearance, and non-flexible, these are, right, going to give more interaction. When the greater interaction will be there, now what happens? Fluidity, yes, decreases. Saturated fatty acids, if they are more, fluidity will be decreased. In case of unsaturated fatty acids, when you look into the tail characteristics here, they are flexible. Why flexible? Flexible in the sense, always it is not like flexible. Flexible is because of this bend. It is not rod like appearance will not be there, thick interaction will not be there, hence less interaction, lesser the interaction, fluidity will become, that's right, more. So higher the fluidity in case of unsaturated fatty acids, if they are more, fluidity will be less in case of saturated fatty acids if they are more, right? Keep this in mind, very very important. Now, so we have understood the three factors. Cholesterol, it's a complex concept you need to understand. So it requires more time to make you people to understand about that. So it's better we are going to learn about cholesterol and FRAP in next class. So in second class, we are going to complete FRAP technique, and we are going to discuss certain questions also, right? So it's very important, students. Today is as an introduction class. I have just discussed about important basic concepts. It's nothing else. So most of you might be knowing about this, but not an issue, right? So this is all about the three factors which are affecting the membrane fluidity. Which one? Temperature, directly proportional, saturated and unsaturated. And again, you know, saturated decreases, unsaturated increases. The third factor, which is long chain and short chain fatty acids. Long chain decreases the fluidity, short chain increases the fluidity. Keep this in mind, right? Now, so we have one question. Just we'll see about that question, right? I'll just write it. It was previously asked question, previous year CSR question it is. So we'll discuss that question, and we'll stop the class here today, and we'll continue our class in next class. Okay, in tomorrow's class, right? So the question was like, a bacterial culture was grown at, we know that it's normally 32 degrees Celsius. We'll consider it as 32 degrees Celsius, and it has been placed at, let us consider it as 45 degrees Celsius. It has been placed at 45 degrees Celsius. Full stop. In order to, in order, in order for bacterial survival, bacterial culture survival, which of the following, which of the following options or options are true? So the question was like, the bacteria was grown in 32 degrees Celsius and it was placed in 45 degrees Celsius. Now, in order to bacterial culture survival, meaning it has to, in 45 degrees Celsius also, it has to live happily. That's the thing. Which of the following statements or options are true? That's the question. Before answering this question, you have to remember two things. The very first thing is, we know that fluidity should not be more, it should not exceed more, and it should not be more rigid also. It should be balanced. Then only the integrity will be achieved, and then the survival of the bacteria will be seen, right? So keeping this in mind, we'll just solve this. So option A was like the very first option was increase in saturated saturated and long chain fatty acids. Long chain fatty acid, Fa is fatty acid. Okay, now, so second question, decrease in same saturated, I mean saturated and long chain fatty acid. Long chain fatty acid. Third question was increase in unsaturated and short chain fatty acids, right? So keep this in mind before answering. You have to think about what these saturated unsaturated fatty acids are going to do with respect to temperature, because temperature is changed from 32 to 45. Keep this in mind and solve. Next, increase in right, increase in saturated and short chain fatty acid, short chain fatty acids. So these are the questions. So try to answer this based on our concept, right? So anyhow, I'm going to explain it. Just try to think which would be the right answer for this particular question. A bacterial culture which was grown in 32 degrees Celsius, which was placed at 45 degrees Celsius. You need to understand the change in degree, change in increasing in temperature, which was lowered the fluidity more. So more the fluidity, if it is more the fluidity, what happens, I mean it is going to increase the fluidity. Higher the fluidity, integrity will be lost. So in order to make it proper integration, what needed to be done, the fluidity should be decreased, right? You have to choose the option such that the factors which are going to decrease the fluidity, those options you need to choose, then only that will become the right answer. I'll explain it anyhow, don't worry, think. Yes, so the right answer for this particular question is see 45 degrees Celsius. Here I'll just explain. 32 degrees Celsius, fluidity will be normal, not too increased or not too decreased. It's happily living at that particular temperature. Now what you have done, you have taken it to the 45 degrees Celsius. So what temperature increase you have increased the temperature, meaning fluidity also increases, because temperature is directly proportional to fluidity, right? So fluidity is increased, so greater the fluidity, phospholipid interactions, they are going to be loosed, and when they are going to be loosed, they are going to separated and the content of the cell will go out. So this is the factor. So you need to bring down the fluidity, then only the bacteria is going to survive. Keep this in mind. Now your intention is to bring down fluidity in order to bacterial survival. Now, decreasing the fluidity, the increased fluidity has to be taken down. To decrease the fluidity, there are certain factors, right? We have already understood. What are those? Saturated fatty acid. Saturated fatty acids are rod shaped, rigid, non-flexible, decreases the fluidity, right? So saturated fatty acid. Second one, unsaturated, it is going to, anyhow it is not going to decrease the fluidity, hence we are going to change this, increase in unsaturated fatty acid is the wrong answer. So second one, decrease in saturated, it should be increased, it should not be decreased, hence we have eradicated second and third. And always remember one thing, the students or aspirants who are able to answer which option is right and which why these options are right and why others options are wrong, that's very important. If you understand that, you'll clear this exam. Now, so we understood 2 and 3 are wrong statements, then answer should be in first or fourth. But in first, what they have given, in first option they have given saturated fatty acid, that's right. So in fourth also they have given for saturated, then how to distinguish now? Now long chain and short chain they have given, no, so which is going to give more fluidity and more rigidity? Of course, that's right, long chain fatty acids are going to decrease the fluidity. So what we need, we need the fluidity should be decreased, right? So the long chain saturated fatty acid is going to decrease the fluidity. That means, so when I look into this option, option 1 is right. That's the reason 4 is wrong, because they have shown short chain. Short chain, again, it increases the fluidity, right? So saturated, it decreases, and again if you increase the fluidity is not going to change. It will be very high in 45 degrees. That's the reason saturated fatty acid increases the fluidity or decreases the fluidity. It is always decreasing the fluidity, because interaction will become more the interaction, fluidity will be less. Longer the chain fatty acids, longer chain fatty acids have more surface area to interact. Now what will happen? Fluidity will be obviously decreased. Excuse me. So this is how the fluidity concept is related with the CSR net. They used to ask not one concept, two or three concepts. They used to combine and they used to ask questions like this. So keep this in mind. So if you have any doubts, then let me know. So after the class, you can just text me, right? So keep this in mind. This is very important for your exam. Based on this, we will be discussing about a next concept called as FRAP in next class, right? Excuse me. So one more thing we are going to discuss tomorrow is cholesterol, which is very important. So this is only 3 concepts we have discussed. About cholesterol and the role in fluidity, we have not yet discussed. Keep this in mind. It's very important. It's very tricky. Most of the students gets confused only in cholesterol relation with fluidity, because cholesterol can increase the fluidity as well as decrease the fluidity. Both, the same concentration of cholesterol can do both. So we'll we'll read about that, learn about that in next class. Okay, so we are going to stop the class here. So we'll continue our discussion tomorrow in tomorrow's class. So the link will be sent as how today how it was sent. So we are going to send it, right? So keep this in mind. Tomorrow we are going to discuss about cholesterol and FRAP. FRAP, very important, right? Based on this, we are going to discuss questions tomorrow itself. Okay, so hope you understood this class. So if I'm fast, then let me know so that I can slow down. So any suggestions can be acceptable. So you can just personally mail. So we know that we have ebiobase16@gmail.com. You can mail it here, right? So you can mail your feedback so that I can improve myself, right? So fine. Anyhow, have confidence with you. So 30 concepts which are very important. Out of 30 concepts, you may you have to write only in part C 25 questions, right? Who knows, up to 20 questions if you get, you will get 80 marks, right? That's the reason. Don't ignore the classes, don't ignore assignments too. I have given an assignment, just follow it. Okay, fine then. So I'll stop the class here. We'll continue our discussion tomorrow, right?